Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 28, going out on September the 26th, which is Mesothelioma Awareness Day, which is a perfect segue to say we have no interviews about that on today's podcast. I'm recording a couple of days early again, mainly because the day the podcast comes out, I'll be doing life sciences interviews in Dublin. I wonder if the rain will follow me from Lithuania, while last week I was doing interviews at Life Sciences Baltics. I think I've been to Vilnius about 5 times now, and the more I get to know it, the more I like it. It's a vibrant place and very green, and certainly up and coming in the arena of life sciences. Unfortunately, this time it was wet as well. If you'd like to hear an interview about how green the city is and about walking there and sustainability, you could always check out our other podcast on walking, which is called Walking Post, and it's at walkingpost.com. Needless to say, quite a few biotech interviews were done in Lithuania, so a few of those will be coming up in the next few weeks. Last week, to mark Childhood Cancer Awareness Month, we had an interview with Paul Romness, CEO of OS Therapies, and we also had a conversation with Immunon's CEO Stacey Lindborg on immunotherapy, COVID, and a few other subjects. This week our guests are Andy Parker, CEO of Step Pharma, about CTPS1 and cancer. And we chat with Michael O'Rourke, CEO of Re-Vana Therapeutics, about eye treatments and a recent deal with Boehringer Ingelheim. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com. which is also the email you can use if you're interested in advertising with us or sponsoring a podcast episode. Of course, first we take a look at some of the news you may have missed over at deeptechdigest.com. Neurosense's drug candidate is showing benefit for Alzheimer's disease patients. There's a possible breakthrough in the development of effective biomaterials, and there are new options for lung cancer research through digital cell twins. Pfizer is acquiring Metsera and its obesity portfolio. There were positive results from a pivotal Alexander disease study, and there has been IND clearance for a long QT syndrome treatment. Eli Lilly is building a $5 billion manufacturing facility in Virginia. The first liquid ambient temperature stable qPCR master mixes are now available, and Calexo Bio has been formed to advance siRNA therapy for cardiovascular disorders. Crimean Congo hemorrhagic fever is in the crosshairs. There's a potential for a new drug targeting BDCA2, and there have been positive results from a phase 1 Huntington's disease trial. And of course, you can read all of these and plenty more at deeptechdigest.com. So first this week, the eyes have it, because we are chatting with someone originally from Scotland, not that far from where I currently live, and that's Michael O'Rourke, the CEO of Re-Vana Therapeutics, which recently signed a huge deal with Boehringer Ingelheim. We'll talk about that in the conversation, and also Ocular Therapeutics and innovative ocular drug delivery techniques from the company that spun out from Queens University Belfast in Northern Ireland. Hi Michael, pleasure to talk with you today on a morning in your home country where it's not raining. To get things started, I wonder if maybe you could tell us a little bit about your interesting background and that of Re-Vana.
Well, I started my career with 3M Pharmaceuticals. I was a drug rep for 4 years all over Scotland and Northern Ireland and so on, and then I moved into Nottingham with 3M. But originally I was a chemistry teacher in Glasgow, and then went into the pharma business. So it was 8 years with 3M, and then I went into the orthopedic business with How Medica back in the 90s. So I was based in London and Hong Kong for a number of years, but then came back and I was with the Alza Corporation. Alza was the world's biggest drug delivery company. J&J bought them for about 12 billion. So opened up their European business and we launched the world's first ever sustained release periodontal or dental product back then. Then they were acquired by J&J. So I came into ophthalmology in 95 with Chiron Vision, based just outside Chiron Vision Europe, just based outside Reading in England. And then so we were acquired by Bausch and Lomb in 27. But during that period, the 95, 96, I led the team that launched the world's first ever sustained release intraocular drug delivery product, which was Vitracert, which was treating a late onset eye condition in AIDS patients, CMV retinitis. So the height of the AIDS back in the 90s, HIV patients would get CMV retinitis, which was a blinding condition in the eye. But we launched Vitracert, which was an antiviral slow release system. So instead of them all getting infusions, constant painful infusions, we had a site-specific implant. So that was the first one ever launched anywhere in the world. So Bausch and Lomb acquired us. I moved to California with Bausch and Lomb for 3 years, running our different divisions. Then I was up in Rochester, New York for 3 years running global pharma. And then Tampa had been the pharmaceutical headquarters of Bausch and Lomb. So I came down here in 2004 to run the US pharma business. So still here today, but so ran that. That was about a $250 million business, had a team of about 150. But we actually then launched the world's second drug delivery device for the eye back in 2005, which is the Reticert product, which is still on the market today. And that was a 32-month delivery system of a steroid, fluocinolone, that treated another eye condition, non-infectious posterior uveitis. So for the first two products ever launched, I led the teams that launched both, one in Europe and then one in the US. The whole grounding in drug delivery came from that, and even prior to that, with Alza, it was drug delivery in the dental drug delivery, and even at 3M, it was sustained release for asthma patients. Instead of taking 4 tablets a day, you took 1 tablet a day, which really first gave me my passion into sustained release and ocular drug delivery, which has never really left me. So Bausch and Lomb were acquired by Warburg Pincus. So a lot of us left. I ran a consulting business. I took over as CEO of Greybug out of Johns Hopkins University for 3 years, and then was introduced to Re-Vana in 2017. So really, the origins of Re-Vana are, it's a spin-out company from Queens University, but what they potentially had in offer was something I'd always wanted to do. So having worked on small molecules in sustained release, the goal that I had was how do you deliver a large molecule, a biologic on a sustained release basis for 6 months or greater, but something that could be delivered in the clinic with no surgery. So it had never been done before because biologics are more complex drugs than small molecules. But equally, any of the technologies that had been beyond the two that I launched and some others that Allergan and others brought out, they were all generic type drugs, approved drugs, and no one even thought about the whole idea of doing a novel drug, which has come full circle that I mentioned. So I joined Re-Vana on the basis of being able to completely change and revolutionize the whole ocular drug delivery market, by looking how to deliver biologics on a sustained release basis to be delivered in the clinic, but also strategically to think about how to take new drugs, pre-approved drugs, into the technology. So that really was my enticement to join Re-Vana, which is a spin-out from Belfast, from Queens University Belfast. It had been founded by 2 professors there. So I joined really with that in mind. Already had some commitment from the financial backers in Belfast. So Cubis is the internal investor. It's like an internal venture capital group in Queen's University. Really amazing group, actually. So they were responsible for bringing startups to fruition at Queen's University. And Queen's, for even later on, a number of years, were the top university in Britain, so above Cambridge and Oxford, to bring startup companies up to an IPO or commercialization stage. So they've founded over 50 different companies. So Re-Vana was one of them. So we had some commitment from them. We had commitment from a couple of the Belfast-based VCs, Techstart, Clarendon, Invest Northern Ireland. They put up a little bit of money, but for someone to come in and run it. So that was the condition. Just a convertible loan note, really. So I joined on the basis of a convertible loan note and took over in 2017. So having been in the States since 1998, I was able, in the ophthalmic sector, I was able to tap in quite quickly into the sort of the US market, but we quickly raised seed money. So we raised about 1.6 million seed money in 2017, and that was Cubis. It was Techstart, Clarendon from Belfast, Invest Northern Ireland, and a couple of private investors. So that allowed us just to get the basics. It's an academic startup. It really came from a PhD thesis, the whole idea. But it allowed us to put some foundational programs in place and start to get our IP sorted out and so on. The long-term goal here was to say, okay, how do you take an academic-based company and transition it into a biotech company? So that was the strategic vision that we had.
Could you tell me about the different technologies that you have?
So we had two different technologies. One's called iLeaf and one's called Ocular. And what was different about Re-Vana is and how we were able to deliver biologics when no one else could was the founding IP was we used photocross-linking. So we used light as a method of preparing the implant and embodying the biologic in the implant as opposed to heat, because heat would just destroy the protein. So the first implant we looked at was iLeaf, and it's preformed, completely ready, but it's photocross-linked with ultraviolet light, and that gives it, so that's the sort of the secret sauce how we do that. And that could then be injected into the eye as a preformed implant. But the second technology is same chemistry, but injected into the eye is a liquid. And when you inject this liquid into the eye, it forms an implant when it goes into the vitreous. So long term, we've worked out how to photocross-link that in the eye, but we do it with visible light. So there's no danger to the retina. So it's ultraviolet light outside the eye to prepare the implant, and then visible light inside the eye. So we had the basis here of two potentially extraordinary technologies, which allowed us to really move forward with the company. But I'd made it pretty clear from the beginning that the strategy behind the company was we wouldn't just be a drug delivery company per se. I wanted this to be truly an ocular therapeutics company. So that meant having two base platforms, two base technologies, iLeaf and Oculeaf, but looking to bring in our own internal assets on the one hand, and externally working with pharmaceutical companies on the other hand. So that's really been a parallel hybrid approach, internal asset development. So we have our own products, but externally, the major pharma companies have got the assets, they've got the therapeutics, but they don't have the means to deliver them. So that's why we've struck up collaborations, obviously accumulating in Boehringer, we'll talk about in the last couple of weeks, but that's always been the mainstay.
So, how did you then move that forward?
We did multiple studies. It was all about trying to show proof of concept that we could deliver a 6-month sustained-release biologic in the clinic when no one else could. Genentech at this time had launched their own sustained-release system with biologics, but it required surgery. So we knew that we still had a very clear pathway forward for Re-Vana as we do today. Queens University have been really, really supportive. So we do contracts on PhDs under Queen's agreement. So we have about 16 in total. With the Boehringer deal, we are going to hire another 5 as well. So we're taking us up to a pretty well established, call it early biotech or emerging biotech is exactly where we are today.
You just mentioned Boehringer, and if you could maybe tell us a little bit more about that Boehringer Ingelheim partnership.
The Boehringer came about as part of our external collaboration strategy. I'd been given a presentation at a meeting, the Ophthalmic Innovation Summit. We were approached by Boehringer after it, and that really started the dialogue. Boehringer, one of the biggest private pharma companies in the world, and we knew that they were looking for a drug delivery partner, really. So they ended up doing pretty extensive due diligence on us from last year, lasted about 8 or 9 months. We agreed a deal on the 22nd of July. It's a strategic collaboration, so it's not a merger, it's not an acquisition, it's a collaboration. And the only exclusivity, as we mentioned in the press release, is target exclusivity. We are totally at liberty to work with other companies, build other collaborations, clearly as long as there's no overlap on the specific target that remains confidential. And that was, a very important part of our negotiations. So it is one company, it's one collaboration, it's a fantastic collaboration. We have now started working on it, but our strategy remains really as is from day one, in that we're looking at 2 different internal assets. We have communicated them publicly. One is a 6-month sustained release of aflibercept, so 6-month sustained release anti-VEGF product. We were also looking at a second generation bispecific product. So there are two internal assets that we're pursuing. Our focus at the moment clearly is Boehringer.
So what would the next steps be?
So really the goal here now is to, make definitely progress with Boehringer. But as we've communicated, we're also looking to raise a 40 million Series B round separately, and that's to advance our internal assets. So really the primary goal here is to take the 6-month sustained release aflibercept product into the clinic. That's primarily the bulk of the 40 million Series B raise, but another portion of that is also supporting many of the other multiple opportunities we have within the company.
Obviously, there are other companies working in this space. What do you think it is that makes what you do different from other companies?
What makes us different and differentiated is our ability to deliver sustained release biologics in the clinic, so no surgery involved. But we also have the capability to go beyond biologics. We can do other molecules. We talk about peptides, large molecules. We've got small molecule capabilities. So there's a whole world of other potential capabilities there. So really building on the know-how and what makes Re-Vana very different is why we're looking at, again, these internal asset development, raise the capital, be successful with Boehringer, and expand collaborations as well. Each year, there's 26, 27 million injections in the eye. The average patient today is getting between 3 and 9 injections a year, depending on the drug that they're given. So what we want to do is really make life better for the patients by reducing the frequency of injections into the eye to 1 or 2 a year. This is what retina physicians want as well. And ultimately, it's what payers will want down the road. There's potential cost efficiencies here. But we do want to break the boundaries here. We want to take ocular drug delivery into a whole new world of capabilities.
Where are you at with your assets?
So we need to raise the Series B. We've done all the basic proof of concept work. So when you bring a drug into a drug delivery technology, you have to do quite a lot of testing to make sure that it's compatible and it's going to work and it's going to deliver. But one of the additional advantages of Re-Vana is that we can customize a therapeutic to our technology. So if you have a target product profile, so let's say you know the size of the drug, you know how long you want to deliver it for, 3 months, 6 months, you want to know what the target therapeutic dose is. We are able to take all this data, put it into a model and say the eye leaf technology is more suitable for this therapeutic, or potentially the Oculeaf is more suitable. So we're able to customize the therapeutics to our own technology, which is a huge benefit for us. And I think from a competitive, it makes us stand out here. We take a lot of time looking at potential internal assets to make sure that they fit our technology. And that's what we've been doing for the last couple of years.
Is there anybody else doing anything similar to what you're doing?
I usually say that I think we're the only company in the world doing our model. But when I started in retina in 1995, retina was hardly anyone was doing retina. When I launched that first technology, Virtucert, for the AIDS patients back in 95, retina was kind of an unknown. No big companies were in it, and it was mostly devices and medical equipment, vitrectomy machines and so on. Retina is going to be $23 billion by 2029, and all the big companies are in it. But the challenge remains that the big pharma companies, the global pharma companies, have only got the therapeutics. They don't have the means to deliver them. So there's a very limited amount of companies that they can actually go to. And we are selected because of our capabilities to deliver biologics in the clinic.
Plenty of opportunity still, and that's good.
Yeah, I mean, the opportunity is just incredible. Populations living older, as you know. As people live older, there'll be more eye disease. There's more obesity. If there's more obesity, there's more diabetes. There's more diabetes. There's more diabetic eye conditions. So the exponential growth of the size, both the prevalence and the incidence of ocular diseases is very evident. We are focused towards the retina, the blinding eye diseases, the macular degeneration, diabetic type eye disease, but we're also interested in glaucoma, which may be a small molecule approach in the future, and other potential conditions in the eye. The Re-Vana technology, although we put it into the back of the eye, the posterior part of the eye, the retina, it can go to the front of the eye as well. So we have that as something else to think about in the future. But as a young company, we're very focused on posterior segment delivery to the retina to reduce the number of injections in the eye. That remains the overriding go as of today. But we recognize that there are multiple other opportunities beyond just the back of the eye, and even going outside of the eye in the future as well. There could be non-ocular applications as well.
Definitely a lot going on at Re-Vana, as that deal shows, and their eye treatments clearly have the capacity to make a huge difference for patients, which ultimately is what it's all about. Next, we're talking about tackling cancers and blood disorders by targeting an enzyme called CTPS1. The company is Step Pharma, and we had a chance to learn about the company and CTPS1 in an interview with Andy Parker, the company's CEO. Hi Andy, thanks for joining us today, and maybe we could start with some details about the company.
Step Pharma is a French company. It was founded 10 years ago, and it was founded based upon the science of an academic clinician at the Imagine Institute in Paris. So as a company, we focus exclusively on developing inhibitors of the enzyme CTP synthase 1. The company's been through various different stages because we really started from a genetic observation. So from that genetic observation, we've been through sort of seed financing rounds, kicking off the program, through all of the sort of chemistry that's required, the financing rounds that have been needed to get us to the point where we've now been in the clinic for a couple of years. The team itself, highly experienced team, decades worth of experience in developing drugs, and particularly in developing drugs in the oncology space. We're backed by a top tier group of European investors, including Kerma Partners, who were the founding investor, but also BPI France, Pontifax, Hadean Sunstone, and others. It's a company that's founded on a very strong scientific understanding of this particular enzyme and the role that it plays based upon human genetics. We operate as a semi-virtual organization, so the team is actually disparate, spread between the UK, France, Switzerland, Belgium, and the US. So it's one of those sort of semi-virtual organizations that relies very much on face-to-face meetings periodically, and otherwise people just driving the programs forward through their expertise and working from home.
You mentioned CTPS1. I wonder if you could go into a little bit more detail about that and its relevance.
CTPS1 is an enzyme that's involved in nucleotide synthesis. So it sits in the pyrimidine synthesis pathway. So the idea that we target nucleotide synthesis isn't novel from the perspective of oncology. It's a known therapeutic vulnerability in cancer. And even targeting the de novo pyrimidine synthesis pathway itself isn't novel in terms of drugs going into the clinic in oncology. DHODH sits in that cascade to develop CTP. Many DHODH inhibitors have been into the clinic in oncology and failed. And any attempt previously to target this pyrimidine synthesis pathway has failed because of dose-limiting toxicity. The reason that we're different and where CTPS1 fits into this is the very final step in that production of CTP is the conversion of UTP to CTP. And here there are two enzymes that can catalyze that reaction of converting the substrate to products. CTP synthase 1, which is our target, and the orthologous enzyme CTP synthase 2. So it's the only step in the sort of 6-step cascade that produces CTP where there are two enzymes. And that creates a point of differentiation that enables us to very specifically target CTPS1 whilst sparing CTPS2. And that brings with it some advantages in terms of trying to attack certain disease states. The original concept comes from human genetics. So Alain Fischer and Sylvain Latour at the Imagine Institute, who are immunologists, geneticists, actually identified a founder population of kids in the UK where those kids failed to mount a response to a viral infection. When they looked at the immune cell repertoire on those kids, it was perfectly normal. When they tried to ex vivo activate those lymphocytes to proliferate, they failed to proliferate. So that explained the phenotype. And then using genetics, they showed the cause of that phenotype was a mutation in the gene that codes for CTP synthase 1, essentially a null mutation. And what made that interesting from a drug development perspective was that that was the only phenotype in those kids. So every other tissue type other than lymphocytes can make use of CTP synthase 2 to make their CTP in order to repair DNA, etc. But in the case of lymphocytes, they're absolutely dependent upon CTP synthase 1. And that was really the genesis of the company. Could we develop a truly selective CTPS1 inhibitor to block aberrant lymphocyte proliferation and get away from the dose-limiting toxicities that have precluded attempts to block this pathway previously? And so that's essentially what we've done in developing dencatistat as a truly selective CTPS1 inhibitor. It makes us the world leader in developing CTPS1 inhibitors, and this is a first-in-class program that's now gone into the clinic, targeting CTPS1 in order to, in a very tumor-specific way, inhibit cancer tumor growth and ultimately kill tumors.
All right, you just mentioned dencatistat. Could you tell me a bit more about that and its mode of action?
Yes, so it's a small molecule. It's essentially a competitive inhibitor to the binding site in the enzyme CTP synthase 1, which is an interesting enzyme because it's actually a tetramer, and you need that tetramer for it to be active. So it's got quite a complex binding site for the nucleotide in order to produce CTP. The molecule itself is over 1,000-fold selective for CTPS1 versus CTPS2, which is absolutely key in terms of the approach that we try to take to target indications that are CTPS1 dependent. Everything to date that we've done in the clinic suggests that it has very good drug-like properties, it's very well behaved, and it's been moving forward quite smoothly through clinical development. And we have multiple clinical trials running now. So really what we're trying to do is to build a pipeline around this single product by exploiting that dependency on CTPS1 in a number of different ways.
Is this an area that a lot of companies or research is being done on, CTPS1 and those pathways?
The idea that you target nucleotide synthesis has clearly been something that people have focused on previously. The concept of specifically targeting CTPS1 is not something that's been explored broadly, at least in terms of publicly disclosed attempts to identify inhibitors. Nimbus Therapeutics were the only company that have really talked publicly about having a program. They expended significant effort in terms of medicinal chemistry, in terms of rational drug design. They also solved the structures of CTPS1 and the related enzyme CTPS2. Otherwise, all the activities that we're aware of are essentially stealth activities. We become aware of companies that are being funded to start to play in this area. So we remain right now sort of standalone in developing dencatistat and really trailblazing, understanding the utility of the mechanism and also understanding the issues that you might have to manage in developing a CTPS1 inhibitor.
Is there a reason why there's not more interest in that?
I think the interest is high. I think the ease with which one can find tractable small molecule chemistry is not there. So we know from our experience that when we ran our initial high throughput screens, the hit rate we got was incredibly low and much lower than you would expect from a conventional high throughput screen. And I think then we were very lucky that we were working with Signature Discovery in the UK to drive those screens and to drive the medicinal chemistry. And they were able to use an empirical make test and also molecular modeling to really take those hits, turn those into credible leads, and ultimately optimize to get us to dencatistat. So I think it's difficult to easily run a screen and find novel chemistry. What we see with Nimbus and what we've seen with a couple of others is attempts to really focus in on the same binding site that we have and end up with chemistry that's relatively similar. So trying to find something novel seems to be the challenge for others, which I'm sure will be cracked eventually. But right now, we are years ahead of anybody else who's coming forward now with novel chemistry.
What kind of diseases, conditions are you tackling?
So we have 3 different studies running. First, study that we started was in lymphoma, and that was really driven by the genetic understanding that lymphocytes have this absolute dependency upon CTPS1 and that CTPS2 cannot complement for the loss of CTPS1. So we started a study looking at relapsed refractory lymphoma patients, and we've been in phase 1 now for almost 3 years. And that study enrolls both B and T cell lymphoma patients. The reason that we have been in phase 1 for such a long time is because we very quickly worked out that we were having an impact on platelets. So we know we have a mechanism-based thrombocytopenia, and it took us quite some time to work through different dosing regimens, build a PK/PD model to understand how to dose the drug to mitigate the thrombocytopenia. So we now dose the drug 1 week on drug, 1 week off drug, and so we see an oscillation in platelets, but that actually manages the thrombocytopenia effectively. And so since then, we've been escalating on that schedule in the lymphoma space, and we're now approaching what we believe would be the dose to enter into the safety expansion cohorts. And importantly, we've seen lots of signs of clinical activity. So we have had complete responses, we have had partial responses as we've gone through the study. The longest duration so far is a patient that's been on drug for 18 months. So we're also building a picture that we get durability with the drug. And in the future, we would expect to focus in on T-cell lymphoma as the opportunity in phase 2. So that's really taught us an enormous amount about the drug, and we've really followed the science as we've gone through developing the story. So intuitively, we felt that going into the solid tumor space was not necessarily an option for a CTPS1 inhibitor because epithelially-derived tissues would be able to utilize CTPS2. So we asked the question, are there examples of tumors that have lost CTPS2, which would create a synthetic lethality and absolute dependency on CTPS1? And the answer to that question was yes, there are. And to our surprise, a very high prevalence of genomic loss of CTPS2 in solid tumor types. So we would estimate that 1 in 10 solid tumors has genomically deleted CTPS2, making those tumors absolutely dependent on CTPS1. And the highest prevalence is in ovarian, bladder and lung, gastroesophageal, endometrial, where we're talking 15 to 20% of solid tumors in those diseases have lost CTPS2. So that enabled us to start thinking about how we would design a solid tumor study, focusing in on using a biomarker to select patients that are deficient for CTPS2. So we worked up an immunohistochemistry assay so we can select patients. We started that study. We're in dose escalation. In the escalation phase, this has been all comers. We're approaching what we believe will be the dose that we would use for expansion. And shortly, we will start looking in ovarian, lung, and endometrial cancer patients that have been selected for the biomarker. So 10 patients in each as a phase 1b study in order to develop an understanding of activity in those indications, but also to develop the understanding of the pan-cancer opportunity here, because this could work in every cancer type where there was a CTPS2 deletion. And so that's really what we would be looking for in the long term with this, as a pan-cancer opportunity. And then the third study that's running, that has just started, we dosed the first patient just last week, is essential thrombocythemia. And again, this is following the science. So having established that we get a thrombocytopenia that's mechanism-based through our lymphoma studies, and understanding the nature of that mechanistic basis for the effect on platelets, we wondered whether this might work in essential thrombocythemia. And having dosed now over 60 patients, we've realized that the only recurrent adverse event we get is the thrombocytopenia. So we have a very clean drug. So now we have taken this drug into essential thrombocythemia with the intention of now trying to normalize the platelet count in those patients. So the drug here is dosed very differently to the way we use the drug in the aggressive cancers in the lymphoma and solid tumors, where we're going for a very high dose complete ablation of the enzyme. In ET, we're just looking to damp down CTPS1 activity to normalize platelets back into that normal range that gets them then away from the issues that they have with the overproduction of platelets, the strokes, the thrombotic events that come from this.
Are thrombocytopenia and thrombocythemia well known?
Yes, well understood. And I think from our perspective, we've looked very carefully to mechanistically understand why are we having this effect. And what we now know is that mature human megakaryocytes, which are the cells that produce platelets, don't express CTPS2. So the progenitor cells do. As they mature, CTPS2 is switched off, making megakaryocytes entirely dependent upon CTPS1. So this is why we have this effect on the platelets. And this is also why this will work very well in the essential thrombocythemia setting, where you've got overproduction of platelets from a clonal megakaryocyte. So yes, it's understood. We understand the mechanism in terms of why the drug works, and we understand how to mitigate that.
Are they conditions or diseases that have lots of people suffering from them?
In lymphoma, that's a more modest number of patients. So if we think about peripheral T-cell lymphoma, then you have about 20,000 patients across the 8 major markets each year. In cutaneous T-cell lymphoma, it's sort of similar numbers. Cutaneous T-cell lymphoma is a condition that people live with for longer, so the numbers increase in terms of the prevalence. But lymphoma represents a much smaller market opportunity than when we think about the solid tumor. When we start thinking about lung cancer, you start thinking about ovarian cancer, endometrial, bladder. Now you get into, there's 100,000 plus patients that you can think about addressing in that population. So from a commercial perspective, from an unmet need perspective, the solid tumor story is obviously the one that drives the value for the company. And then essential thrombocythemia is also interesting because that's a condition where people are diagnosed in their 50s. If the platelets are managed, they live a perfectly normal lifespan. So you've got an opportunity here where you need to be treating people for 20, 30, 40 years. So it's essentially a chronic therapy. So the prevalence there is actually very high, representing a very interesting opportunity. It's currently treated with hydroxyurea, which is kind of cheapest chips chemotherapeutic drug, but 25% of patients fail ultimately on HU, and it's that portion of the patient segment that we would target with this as a novel second-line agent to really bring about good control of their platelets. So huge opportunity in solid tumors, an interesting opportunity in ET, and a smaller space in lymphoma. And there we choose to focus on T-cell, primarily because the B-cell lymphoma space is very crowded with CAR-T, with biospecifics, changing the way that the treatment paradigm is really put together. Whereas in T-cell lymphoma, there are no approved second-line agents in peripheral T-cell lymphoma in Europe, only a couple in the US, neither of which are particularly effective. So there's a wide open opportunity, clear path to market in T-cell lymphoma. And in fact, we have orphan drug designation for both peripheral and cutaneous T-cell lymphoma. So that will also help us as we move forward with the agencies.
Could you tell me about some of the clinical trials that you're doing?
Yeah, so in terms of the clinical trials, the lymphoma clearly shows clinical activity as we've gone through escalation. The intention would then be to move into a phase 2, which is currently agreed as a Simon 2-stage design, where we would open up arms in peripheral and cutaneous T-cell lymphoma and look at around 30 patients in each initially. It is also a space where, with limited data, you could think about moving relatively quickly into a broader pivotal study. It still has the opportunity for accelerated approval as you enroll the majority of patients. So these are the kind of discussions that we'll be having with the agency as we get towards our end of phase 1 meeting and moving forward on into phase 2. In the solid tumors, the idea here is really to use the phase 1b expansion cohorts to demonstrate activity across 1, 2, or 3 of those indications as we look in ovarian, in lung, and in endometrial. And then ultimately, we need to think about what the phase 2 design will be and whether we focus in on a given single indication or whether we actually broaden that out and try to focus on several in a basket type study, for instance. So that's still something that we need to consider, and it'll be driven a little bit by what we see in the phase 1b. Interestingly here, for each of these indications, we have to develop the immunohistochemistry assay separately. So there's a body of work that needs to be done. So far we've done that for 3 indications. We could think about doing additional ones. As we're doing ovarian and endometrial, we've thought about building a gyn-onc franchise so we could next focus on cervical, where there's a reasonable prevalence of CTPS2 loss. But that's very much a work in progress in terms of exactly what the phase 2 looks like. ET itself is very straightforward. So right now we're going to recruit 20 patients into that study. If that looks interesting, we'll expand that out to 50 patients. And at that point, we could probably move quite quickly into a pivotal study. So we could, by 2027, be into a pivotal study in ET. So that may be the program that moves the fastest along the regulatory path compared to the other 2.
For those trials and the indications that you're working on, what would you define as success, and not just for the company, but I guess ultimately for the patients?
In T-cell lymphoma, I think actually it's a relatively low bar to develop something that brings greater success and benefit to patients than is currently available. The second-line agents that are available in peripheral T-cell lymphoma have about a 30% effect and a mean PFS of about 4 months. So, really we'd be looking to try to get at least a 40% effect in that patient population, and ideally get a duration of action that gets you to a year or beyond. And then we'd really be changing the bar for those patients who have relapsed in that condition. In the solid tumor space, I think it would just be fantastic to demonstrate that our CTPS2 null hypothesis holds true and really begin to show the pan-cancer opportunity here and to develop this in as many indications as possible. But of course, that's going to be finance dependent. So we need to demonstrate the POC in at least one of them and start building the story for the pan-cancer approach. Similar to things we've seen with HER2, etc., in building that out slowly but surely to show the true potential and the impact that this could have. And in ET, it's proof of concept in the first 20 patients and really demonstrating that we've got a really clean agent that could make a difference for those patients that are looking for the second-line agents, because the currently approved second-line agents all bring with them some safety concerns. So there's a real opportunity here if we can show that we've got a safe drug that actually brings platelets back into the normal range and maintains them there. So those would be the sort of success parameters in terms of the clinic. And of course, from a company perspective, building out the company to enable us to really move forward into pivotal trials, ensuring that we have the investor support to enable us to do that. Those, I think, are the sort of short-term and even the long-term success factors for the company.
Speaking of that, how is the fundraise going? I guess it's usually these things are ongoing. And also, are you looking to go it alone, or are you looking for partnerships?
Financing is always tough. That's never an easy thing to do. And I think even today, it's even worse. I mean, if you're a preclinical company, I think it's a real challenge. I think there's a feeling of looking for clinical stage de-risked assets as a first port of call for many investors now. And I think there's also a resurgence in interest in small molecules, which is good for us. So the heydays of cell and gene therapy and RNA therapies seems to be on the wane. Antibodies and small molecules, are always there with their different pros and cons. But I think small molecules are definitely making a comeback. From our perspective, I think we've got a very strong scientific story. We've got a very clear rationale. We've got good signs of clinical activity in the lymphoma study, and we're on the cusp of generating that in the solid tumor and in the ET study. We are in a financing mode. You're right, it's almost always continuous. So, we're getting close to the end of raising the next round, and hopefully we can announce that sometime relatively soon. That we are well financed to get ourselves to deliver, some of those short-term milestones around proof of concept in different studies.
Has what's happening in the US affected fundraising any? I know there are different areas and there have been cuts with vaccines and that kind of thing, but does nervousness in one market kind of spill over to others, or does it mean that because it's shaky in the US, they're looking to invest elsewhere?
I wouldn't say it's had a significant impact. I think some funds that are heavily exposed in the public markets as well as private investment may have modified their investment position as they've seen markets be a bit shaky. But in general, I find the discussions that I've had with US investors versus European investors hasn't shifted enormously. I just think the risk appetite is slightly different now to where it's been. I think the idea of platform companies is not something that's de rigueur anymore. And as I say, I think the cell and gene and RNA-based therapeutics are also under greater scrutiny, particularly gene therapy, and with the things that you've seen more recently with Sarepta and others. Just trying to get past that sentiment is maybe more difficult. So I like to play on the fact that we're a small molecule with a very clean mechanism, very clean profile, clear story in terms of how we're approaching the different conditions, and hope that resonates well with the investors that I talk to.
You mentioned the things that you're working on, which is quite a lot, but are there other things in the pipeline?
So we're very focused on just building out the pipeline around this one product. There are other compounds that sit in the wings. We don't currently have any plans to particularly develop those. There's always been the idea of going into, other sort of peripheral indications. The immunology space has always been interesting, GVHD. These things might require us to develop a different compound. But at the moment, all the focus is really on maximize the value of denicadstat, do what we can across all three of those clinical trials, which I think, in the end, we probably will have to also start making choices about how we prioritize there, because it's an awful lot happening with one molecule, and there will be a need to sort of separate out the stories and decide where we focus ultimately, because in the end, if we're moving into pivotal studies, then the finances required to drive all of them, will be quite significant.
Yeah, it's very easy to overstretch yourself and get kind of diluted by doing too much, so it makes total sense.
Yeah, in the end, it's difficult with a small team to be driving everything. So I think we'll also be looking at how can we creatively work with others, other opportunities to license out based upon indication, other opportunities to do licensing deals that relate to specific geographies, but always being mindful of the fact that we're built around one asset. So in the end, some of those things are complicated to do, but we will certainly be looking at whether there are ways to carve out something, work with somebody else, spread the risk, spread the cost, etc.
Is there anything else that you wanted to cover that we didn't mention?
It's a first in class. It's a really interesting mechanism. It's shown really good signs of clinical activity already in the lymphoma space. And I honestly believe that this is a molecule and a mechanism that has huge potential across multiple indications, from those aggressive cancers to the benign hematology indication of essential thrombocythemia. So I think this is exciting times for us and exciting times in terms of demonstrating what we can do with this molecule.
Yeah, and as you mentioned earlier, as the only one doing this and being so far ahead, you're not looking over your shoulder all the time either, so that's good.
No, we're not looking over our shoulder from a mechanism perspective, but oncology is hugely competitive, so we're always looking at what else is being developed in that space, particularly in the solid tumor space, less so in T-cell lymphoma and ET. So we always need to demonstrate that we're differentiated, that we bring something new. I think the clean profile also helps in terms of the only thing we're managing is the thrombocytopenia, which we can manage well. So we're a great combination partner for many drugs down the line in the future when you think about how we explode out from monotherapy in relapsed refractory patients to what we can do when we combine with other drugs.
Another in-depth one there, and now you know all about CTPS1. If it should come up in a dinner conversation this weekend, you could always just shoehorn it in. Anyway, that's it for this week's podcast. I guess I have lots of interviews from Lithuania to edit for the coming weeks, so I'd better get to that. once I get back from Dublin, where I'll have more interviews to get through. It never ends. So until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.